Okay, so welcome to our first online class lecture. It is March the 16th, and you are in the medicinal chemistry course. Um, so today our goal is to look at the packet. The packet should be uploaded on your Canvas. I think last week before spring break you were given a hard copy, and it's for POGIL Activities Part C. And you were to go through and you need to choose your disease, a drug target, identify a bioassay, find a lead compound, identify structure-activity relationships, identify the pharmacophore, propose structural changes to improve the target interactions, pharmacodynamics, and improve pharmacokinetic properties. Okay, so you get to go ahead and choose your disease and drug target. And so if you could just go ahead and fill all that out, that would be good. What I'm going to do is I'm going to take this 20 minutes, and I'm going to share with you the notes that I have taken from the reading and what I find is important. And you can take notes from that, and then that should help you for when you have your quiz on Wednesday. We'll have a Wednesday quiz. And we'll have your typical 10 questions, and it'll be worth 10 points, okay? And it'll come from this. All right, so first, your goal, once you figure out what your drug target is, and you identify the bioassay, your goal as a chemist is to find a lead compound. Okay, so what is the lead compound? This is an active principle, that's like a component, isolated from a natural source or a synthetic compound prepared in the laboratory. So we find a lead compound, and then we will make changes to that. So in drug discovery, you have to choose a drug target, and this is where I want you to choose your own drug target as an example and kind of take it through the steps. So you figure a disease model. So find something that's interesting for you. I mean, for me, it was cancer. So I ended up doing prostate cancer. Okay. Now, I've also been on an obesity project. And when you pick a drug target, a lot of times in the industry, you follow the numbers. In the developed world, the how much money that drug molecule can make. A lot of times, like migraine, obesity, flu, cancer, cardiovascular diseases, these are diseases that tend to have more money. So the sad thing is, a lot of the in the undeveloped world, some diseases will not have so much drug focus. And then you find your drug target. Okay, so hopefully you can find the receptor. Maybe get an X-ray crystallography structure of that. It could also be an enzyme inside this cell, or it could be a nucleic acid. So you also, once you figure out what your drug target is, you find your disease. Here's your disease, and then you find your drug target. So my disease was prostate cancer, and my drug target was the estrogen receptor beta, and then from there you can decide whether you have an agonist or an antagonist. I had an agonist, and so that turned it on, right? It turned on the receptor, and but you can have an antagonist. So you get to kind of propose those. And then the next thing you want to do is define your bioassay. So another thing I want you to know is, well, first of all, before we go leave drug target, let's talk about the receptor. Another a word that I think you should know is an orphan receptor. What is an orphan receptor? It is a new receptor or targets without lead compounds that interact with them. This is usually the driving force for combinatorial chemistry. Okay, so combinatorial chemistry is when you make just like maybe 100 compounds, and then you put them in a 96 well plate with a lot of different targets. Also, it's very similar to parallel synthesis, and then you try to find some kind of binding, and that binding determines whether you have a target or not. The other thing you want to look for in your drug target is selectivity and specificity. Okay, so this will decrease your side effects. A lot of time your side effects will be very dependent on your pharmacokinetic properties, and so it's very important that you're developing both your pharmacodynamic properties along with your pharmacokinetic properties. You don't just do one and then focus on the other. Okay, now we're going to go to the identify a bioassay. So if we identify a bioassay, you have two. You have an in vitro and you can have in vivo. And I want you to be able to define these and talk about the advantage and disadvantage. So in vitro, what is that? That's usually isolated cells. They could be tissues, isolated tissues. They could be enzymes. They could be receptors. Okay, so they're actually isolated. One of the advantages is it costs less money. And you can get the results fast. So we used to get like 2 to 3 day turnover for our in vitro. In vitro, I think of it as a test tube, in vitro assays, and we get the KI data, and then that would make decisions. Okay, for in vivo, these are your animal. Okay, both tests. So if you get a drug candidate, they will have to do well in both the in vitro and the in vivo assays. They are they cost more money. Now the other thing that you want to think about too on the in vivo assays are some drugs that have showed, like, so you've taken a lead compound and you put it in, let's say, like rabbits, not rabbits, but okay, so let's take penicillin. Penicillin, an example. Take penicillin, penicillin methyl ester prodrug. Okay, that will be hydrolyzed in mice and rats, but not in rabbit, dogs, or humans. So when you're thinking about in vivo and animals, a lot of times you have to try all different species. A lot of times they'll use transgenic animals. So what are transgenic animals? These are animals where the genetic code has been altered. They'll replace, it's really kind of fascinating, they can replace the like some mouse genes with human genes. And so then what happens is the mouse receptors will actually produce human receptors. Now these are more expensive and the data costs more, so usually, and they also cause animal suffering. So animal suffering, and they're really expensive. Okay. The other thing I wanted to say, going back to the in vitro assays, is you also can have genetic engineering, and you can incorporate a gene for an enzyme into these fast-growing yeast cells. An example of that would be the HIV protease, E. coli. Okay. And then the other one would be, so it gets expressed in the E. coli. Another one would be the Chinese hamster ovarian cells. Sometimes you'll see that as CHO cells. And this will express a cloned receptor on the cell surface. And those are basically in vitro assays. So that describes your bioassay. And the whole goal is to be able to evaluate a lead compound, to find a lead compound, something that's biologically active, and then you're going to want to make structure-activity relationships with this structure. So what is that? Well, excuse me, sorry. The other thing, before we go into the structure-activity relationships, go through your book and identify the ways a lead compound can be determined. So there are people that just specialize in screening natural products, looking through the plant kingdom, studying microorganisms, bacteria and fungi, marine sources, animal sources. They look through venoms and toxins. So your book has pages and pages of ways to identify a lead compound. And once a lead compound is found, then it's a starting point. So that becomes your starting point for your drug system, drug design. And the next two chapters in your book really focus on strategies for drug design. And that's what I really want you to get to on your molecule. So once you find your drug target, you find some molecules, you can use the mole inspiration. I gave you the website on the last page here, www.moleinspiration.com, and you can draw your molecule in there, and you can do different iterations, and that will tell you biological evaluations of your properties. And what I'd like for you to do once you define this is to make those iterations, but I'm going to show you what iterations to make. And so your homework from this 20-minute video, as we do have a 50-minute class, will be to go through your book and set up a chart like I'm going to propose for you. And I want you to be able to attach that and send that by an email. So we'll get to there. And those are going to be design strategies for drug design. So what are your goals for your lead compound? You want good selectivity. Okay, you want good level of activity towards the target. You want minimal side effects. You want something that can be easily synthesized, and you want something that's chemically stable. You want something that's non-toxic. That's why we think about phase 1 and phase 2 metabolism. You want something that has acceptable pharmacodynamic properties. Okay, so our goal is to optimize the interaction of the drug with the target. So these are pharmacodynamics and pharmacokinetic properties. We're going to tackle them at the same time, and we want to give them equal priority. And so you're going to try to design a structure-activity relationship. And this aim is to identify those parts of the molecule that are important to biological activity. And then you want to identify those that are not important for the biological activities. Many times you'll have an X-ray crystallography spectrum that can be used to understand the target-ligand interaction. Sometimes you're not given that. We weren't able to have that until 2006. One thing that did come out, China did get an X-ray crystallography structure of the coronavirus. So when you have the crystal structure, that kind of helps you understand that biological target, and it makes it easier to find drug molecules that can interact with that target. Okay, so our goal here is to recognize the functional groups. We want to understand their bonding. So this is for PD, pharmacodynamics. We want to understand their intermolecular forces, interactions towards the binding site. We need to define those that are important in the binding interactions. We will remove functional groups, or we will alter functional groups. And then by doing that, we decide whether something is essential or not. That's how you would determine whether something's essential. Okay, so let's make a chart. So I want you to go through your book, and I'm going to get your chart started, and this is what I want you to make for yourself, because I'm going to let you use that chart in your quiz. Okay, and I also want you to have that chart for your POGIL points. Okay, so in your chart you want to write functional group. All right, so you have a chart there, and then you're going to have the type of bonding, and then you're going to have the analogs. Okay, so this is the strategy to make, and then over here you can write notes. Okay, so the first one, you can go through your book. What page is this? I believe this is after finding a lead compound. This is going to be chapter 13. Okay, so we're looking at chapter 13 and 14. So we're going to combine these two. And the first functional group, and it's on page 224, I want you to write alcohol and a phenol. And if you need to, you can write an example of that. Okay, so an alcohol or a phenol. Okay, so that's what those functional groups look like. What type of bonding does this do? This does hydrogen bonding. It can be an OH bond acceptor, or it can be a hydrogen bond donor, because it has the hydrogen there. It also has the lone pairs on the oxygen, so it can be an acceptor or a donor. And then the analogs you would make, you could replace it with an O-methyl ether or an O ester, methyl ester. Okay, so that's on your molecule there. So this is a methyl ether, and this is a methyl ester. And what does this tell you? This will let you know whether that extra bulk will, it should hinder your hydrogen bonding. Okay, so you'll learn whether it's hydrogen bonding or not. And these are easy to make synthetically. Okay, and then the next one, you'd have aromatic rings. And they are planar and they're hydrophobic. They interact by van der Waals interaction. Okay, and so they're going to detect, they're going to lay flat in a hydrophobic pocket in that target. Okay, so what analog would you change? You could replace it with a cyclohexyl ring. And a cyclohexyl ring is going to lose that planarity. Okay, it's not flat. And that would let you know. The next one, alkenes. And keep going. And you go through your book and you find these. I've got ketones and aldehydes. We have amines. We have amides. And have I done any more? Let's see. I've also done so we got amines and amides and ketones and aldehydes. You can do quaternary ammonium salts. Now remember, this chart is for you to help you do your drug design. So if you need to show what a quaternary ammonium salt is, put an example there. And then also I have carboxylic acids. Okay, so if you have a carboxylic acid, you can make those changes. All right, well, start making your chart, and if you have any questions, you can add them to the discussion board in Canvas. And if you send me anything that you need me to grade or look at, you can send it as attachment by email. And I want you to write CHE 335. And then in case this would be the Pogel C, and then your name, and then keep your last and your first name. And I apologize for all the background noise.